All right, so this is my slideshow on reproductive basics. That is the basic stuff you need to know before we get into the details of the reproductive system. We're going to identify the gonads. We're going to talk about gametes and meiosis, and we're going to briefly throw in a little bit on genetics and heredity, and that'll set us up for everything else we need to do. Okay, so I like to start all of my body system units with this question of why do we have this body system? What has it ever done for us? The question of why we have a reproductive system is actually a little bit harder than most of them, because most systems, it's pretty easy to see why we need that system to survive and thrive. It helps maintain homeostasis, or it helps us adapt to some important condition or something like that. The reproductive system really throws us off.
It helps you survive too.
How?
Without the reproductive system, there'd be no person.
Okay, yeah. So why we reproduce really is to survive as a species. The essential answer to why we reproduce, and I always like to say this because it's just kind of shock value, but we reproduce because we all eventually die. And if we didn't reproduce, the human species would die out. My particular genes would die out if I didn't have children. And I think my genes are cool, so, I want to have cool children. Okay. So the question, though, that begs the slightly more interesting question. Okay, but why do we reproduce sexually? Okay, sex is fun, but it's also a real hassle to spend most of your teens and, in my case, 20s finding a reproductive partner and that's a big hassle. If I were able to just bleb off a little mini me who is an exact copy of me and raise that kid by myself, I could reproduce without all of that hassle. So does anybody have any thoughts on why?
Because if you kept reproducing the same person, you would all die of the same thing, and you would have all the same mutation.
Yeah. Okay. Great. Y'all are making my job easy. Okay. So from an evolutionary point of view, the number one reason we reproduce sexually, as some of y'all have said, is genetic diversity. Okay. If I just made exact copies of myself, there would never be any mixing and matching of genes, and if I had some deadly weak gene in there somewhere, then me and all my progeny would die off. On the other hand, if I have, say, a weak liver gene, but my wife has a strong one, and I have a stronger gene in some other area than she does, then we can compensate for each other and have a kid who's stronger in both areas. Okay, they could also inherit both of the weak genes and be a complete wimp and die. But you run that risk. But you have the ability to build better genetic people by mixing and matching genes every generation. Another one that I like to point out is two parents are better than one. Okay, not to say that single parent families can't survive, but the general data suggests that if you have one parent to watch the kids while the other is going out and foraging for food, or one parent to watch the nest while the eggs are developing, and the other one goes off and catches worms, or whatever parent setup you have, that provides a better chance for the offspring to survive, and so it's that's an advantage of reproducing sexually, is you have to have two parents at least to get it started. Now, it's always possible for the dad to kind of fire and forget. It's in some species possible for the mom to as well. I mean, I think some species of fish, the mom swims along dropping eggs and the dad swims along dropping sperm, and they never see whether it even created an offspring or not. Sometimes I think I'd prefer that, but no. No, I've had a wonderful experience raising my children. Okay. The third one, though, is that there is a social bonding function generated by sexuality. Okay. The fact that we spend all that time in our teens and maybe 20s and maybe 30s for some people, trying to hook up with other people means that we have a lot of social interactions with people. The fact that some of us end up successfully mating for life gives us a built-in social partner for that period of time. Those things, you can't underestimate how beneficial that is to humans as a social species. So that's another big thing. And, plus also sex is fun and a good release of tension and energy and all that. Or at least I remember it being back when I didn't have kids. Okay. So we've established we need to reproduce, and there are advantages to doing it sexually. So what's necessary to accomplish that? There are basically 4 steps to reproduction. Step 1 is to generate male and female reproductive cells. So we have what are called primary reproductive organs, or gonads. That's going to be in this little lecture plus the next couple of classes. Step 2 involves bringing those reproductive cells together. Okay, sperm meets egg, generates a baby. That's the step that we think of as having sex or sexual intercourse. And that's the topic a couple of classes from now. The organs that aren't primary reproductive organs, penis meets vagina, that sort of thing, all take part in that. Step 3, dad is left out of in the human species, is the development and incubation of the baby. Again, in some species of animals, mom's left out too. She can just lay an egg somewhere and walk off. But for mammals, at least, we have to carry our young, and so that's a thing. And we're going to talk about pregnancy and human development in our very last class period. Finally, step 4, nurture of the new individual, which starts but definitely does not end with these marvelous things called mammary glands that are what make us mammals. I mean, the word mammal means something that has a mammary gland. So if you want to see this as kind of a flow chart, gonads have to produce gametes. That's going to be our first major topic. Intercourse has to bring the gametes together. That's going to be our second major topic. We have to incubate or nurture the conceptus. That's a fancy Latin word for the baby to be. And then, mammary glands feed the infant, parents nurture for, I put question mark years. There was a time in human history where we turned our kids out of the house at age 13 and said, go have your own life. Now it's at least twice that long. Now I know people whose kids have come back from college and are living in the house again. So, it gets longer and longer all the time. Okay. So this first step is what we're focusing on today, gonads making gametes. What are the gonads? For the male, it's the...
I don't even know the proper term. I was just going to say the balls, but testes. Testes.
Yes, testes.
I was like,
The cojones, yeah, okay. No, for female, it's ovaries, yeah. Yeah, we have this culture in America of having lots and lots of alternative names for testes, and they're used as a symbol of, I guess, chutzpah. You know, that person's got balls or whatever. That's a ballsy thing to do. I had a friend in college, a very feminist friend, who tried to get the phrase "that girl's got ovaries" to be popular, but you don't hear people saying that around these days, so I don't think it worked. Okay, endocrine organs. The gonads are endocrine organs, so naturally they produce hormones in response to a couple of tropic hormones. Anybody remember what they are?
FH.
You're mashing them up there. It's FSH and LH. Okay, that's fine. The male hormone, of course, is testosterone. What I have plenty of in my household of 4 boys. And the female is a little more complicated. For the first half of her monthly cycle, she makes mostly estrogen. For the second half, she makes mostly progesterone. Okay. But gonads are not only endocrine organs, they are also reproductive organs, and so they produce gametes in response, always in this case, to the hormone FSH. So the male gamete, the male reproductive cell, we all know as sperm, or as my boys used to say it when they were little spermies. What does anybody know the fancy full term for them?
I think
Spermata something. Yes, that's good. Spermatozoa. Singular is spermatozoan. It comes from the Greek spermato, meaning to give rise to something, and zoe, meaning life, like animal life, like going to the zoo or studying zoology. So this is that which gives rise to life. You can tell it was a man who came up with these names. Okay. The female one is oocyte, from Greek oos, meaning egg, and cyte, meaning cell. So put those together, we get egg cell. Not nearly as self-aggrandizing as that which gives rise to life, but just as, or perhaps more important. Okay, so what are gametes? Gametes are reproductive cells, which means they take place in a formula where the male makes millions of spermatozoa, the female makes a single oocyte, and if all goes well, 1 spermatozoan plus 1 oocyte gives you 1 zygote, which is a fertilized cell, which is the first stage of a conceptus. It goes from zygote to blastocyst to embryo to fetus to all sorts of things. Now, if you look at this carefully, we've got an equation where 1 plus 1 equals 1, and the math doesn't quite work out. So technically, to make 1, you need to add up half plus half. So this only works if in some way a sperm is half a cell and an egg is half a cell. Now, you may say, that's cute, Dr. Camp, but what do you really mean here? What I'm driving at here is there's one organelle that most human cells can have only 1 copy of. And that's what? Right there in the middle of the cell, the nucleus, right? Okay. And so a gamete has to have half a nucleus. Well, we're not going to have like a little half circle in the middle of the cell. So really, it's more that it has to have half of the stuff that's in the nucleus. And what's in the nucleus are your chromosomes, the things that contain your genes that ultimately make you able to live. So a gamete then is a cell that has half the chromosomes. Okay, so gametes are half cells with half the total number of chromosomes that will eventually come together to make whole cells if they're lucky. Okay, but there's a problem in getting all this organized. Let me try to explain that. Let's say a sample organism has 4 chromosomes, A, B, C, and D. If the chromosomes were just randomly divided, the sperm could get A and B, and the egg could get B and C, okay? And if a sperm with A and B fertilized an egg with B and C, what's the problem? There's no D, okay? So whatever life-enabling genes are found on the D chromosome just aren't there. This baby's going to die. So nature's solution is for every somatic cell, that is a non-gamete, to have 2 copies of every chromosome. If every somatic cell has 2 A's, 2 B's, 2 C's, and 2 D's, then when we split them up, every sperm and every egg can get 1 A, 1 B, 1 C, and 1 D. And when they come back together, the fertilized zygote has 2 A's, 2 B's, 2 C's, and 2 D's. See how that works? Nice and neat solution to that problem. Now, we get tired of saying 2 copies of every chromosome in some cells and 1 copy of every chromosome in another cell. So geneticists have made up some shorthand terminology. We call the number of unique chromosomes an organism has N. So the organism we've just been studying, this A, B, C, D, would have A, B, C, D, 4 chromosomes. So N would be what? 4, right? Okay. Humans have N equals 23 with a little asterisk that the 23rd chromosome is our sex chromosome. So some people have 2 X's, but some people have an X and a Y. Somatic cells all have 2 times N chromosomes. We have 2 copies of every gene. So for our sample organism, that's 2 times 4 is 8. For humans, that's 2 times 23 is what? 46. Okay. So that little kit 23andMe really should be called 46andMe because they're really checking all 46 of your chromosomes. 1/2 cell gametes have 1 times N chromosomes. That's called haploid. So that's 4 for our sample organism, or 23 for us. With me so far? Okay, let's try it. Okay, so we still haven't answered that key question. How do we make sure each gamete gets the chromosomes it needs? Let's say a somatic cell in this pretend animal that we're looking at has A1, A2, B1, B2, etc. If we just divided the chromosomes randomly, we might pick that one, that one, that one, and that one. And once again, we'd miss out on the D chromosomes entirely. Problem, right? So we need a way to specifically pick one of these 2, one of these 2, one of these 2, and one of these 2. So we need a type of cell division that sorts out our chromosomes. If you remember from A&P1, cell division, somatic cell division, uses mitosis to sort out its chromosomes. Y'all remember that? Okay, there were 4 stages, PMAT, that sort of thing. In gametogenesis, we're doing a special kind of cell division called meiosis. And I'm going to try to show you the differences between those in just a second. Okay. In mitosis, remember, we want to make genetically identical copies of our cells. So we start by duplicating our DNA in S phase. See, there are copies of all of our DNA. And then we divide the DNA evenly so that 1B2 goes here and 1B2 goes here, and the same for all of the rest of our chromosomes. So this somatic cell is exactly equal to this other somatic cell, at least as far as the genes are concerned. Okay? That's what we want when we duplicate most of our cells, is to make an exact copy. But in meiosis, we want to make gametes with half the normal number of chromosomes, and we want to make sure that they get the right half, the correct half. So again, we start with S phase. Okay, there we go. We've duplicated our DNA. But this time we group together what we call homologous chromosomes, the chromosomes that go with each other. Okay, and then we separate those pairs in a process called meiosis 1. Okay, so A2 goes this way, A1 goes this way. Okay, see how that works? That's kind of cool. Now we know that each secondary germ cell has one of the types of each chromosome. Starting with that as the starting point for meiosis 2, we can then divide up the germ cells so that 1B2 comes this way and 1B2 goes this way, and we end up with 4 gametes, each of which have half the number of chromosomes, but each of which have one of each letter, if you will. 1A, 1B, 1 X or Y. Cool, right? Okay. So what just happened? We produced 4 not genetically identical half cells. Each got 1 orange and 1 green, but some got a dark and some got a light. Each gamete also got either a red X or a blue Y. Okay, you can't be both. You have to pick one or the other. And the most important thing to note is the chromosomes were independently assorted. Just because one cell got A1 doesn't mean it also got B1. Okay, it might have gotten B2. And so sometimes the ones go together and sometimes they don't. That's independent assortment, and it helps randomize the genome every time we do this. So now let's think about what happens when these 2 haploid gametes come together. We've got gamete 1 here who's got the dark A and the light B and the X. Gamete 2 has the dark A and the dark B and the Y. They come together, and we get a zygote with 2 A1s, a B1 and a B2, and an X and a Y. So we've got some genetic diversity over here, and we've got some genetic sameness over here. There's no guarantee that sexual reproduction is going to produce genetic variety. Sometimes, mom has one gene and dad has the same gene and they come together. Okay. So when that happens, the new zygote will again have 2 green and 2 orange, one from the sperm, one from the egg. It might get 2 dark green or 2 light green or one of each. So what happens if the dark green and the light green are different? What if one of them codes for brown eyes and the other one codes for blue eyes? This was a problem that geneticists had a long time ago when they were thinking about all of this, 1800s or so. One of the options they thought about was, well, if you have a dark-eyed parent and a light-eyed parent, the child will be in between. We know that doesn't happen. Otherwise, we'd all eventually have gray eyes. Okay. Option B is that we always take after one parent or the other. One parent has stronger genes and the other has weaker genes. We know that's not true because sometimes we have dad's eyes and mom's nose, or some other odd mix mash of things. Option C is there's a tug of war between the 2 genes until either brown or blue wins. The reality is actually kind of close to that. It's sort of like B and sort of like C, but not quite either one. Okay, so hold on to that thought for a minute, come back to it. The other thing is the new zygote will have a mix of the blue and not pink, but red is how I colored it. Human females, in case you didn't know this already, are XX. So all egg cells are going to have what chromosome? X, right. Fairly simple. Human males are XY. So half of sperm cells are going to have an X chromosome, and half of sperm cells are going to have a Y chromosome. So one sperm plus one egg, we're always going to get an X from mom. Dad can give us another X, which makes a female, or a Y, which makes a male. So there's a 50% chance of each child being a boy or a girl. And it's the sperm that decides, so if there's a problem with the gender of the kids, blame the father. Sorry, Henry VIII, who is famous for having his wives killed or imprisoned or whatever because they had the wrong gender children. Turns out he just shot a lot of X's. Okay. So back to that question of dark green versus light green. When mom and dad's traits are different, which one do you inherit? We're going to take a little journey to a garden and see if this famous garden can shed some light on this. The garden belongs to a monk named Gregor Mendel back in the 1800s. And Gregor Mendel, when he was put in charge of his monastery's garden, or at least a portion of it, decided he was not going to just randomly plant things. He was going to do some science experiments. So he took a green pea and crossed it with a white pea to see what would happen. And what he got was that every pea plant he planted produced green peas. Now he could have stopped there and said, okay, well, green is stronger than white, so green must always win. But he didn't. And what's really cool about Mendel is that he then took two of these and crossed them. And what he got in the next generation was 3 green for every 1 white. So the white came back. And he thought about this and thought about this, and finally, somehow miraculously came up with the right answer, the answer we still use today, the answer we call, in fact, Mendelian genetics, which is that there are some genes that dominate other genes. So the dominant genes he writes with a capital letter. So capital G is green. And the weaker genes he writes with a lowercase letter, so little w is for white. What he reasoned out is that the peas he started with, one of them had 2 copies of the green gene, and one of them had 2 copies of the white gene. When he crossed them, the only possibility is to get a green and a white together in the same plant. And when green and white are together, because green is dominant, green always wins. So all of this generation had green peas. But when you cross a green white and a green white, there are 3 possibilities. You could get 2 green, you could get a green and a white, and you could get 2 whites. And as long as you had the dominant trait, you kept getting green peas. But if there was one that only had the weaker gene, the recessive gene we call it, because it sort of recedes into the background, that recessive gene came out when it was the only gene there. Okay, so what does this tell us about human heredity? Your phenotype is what is expressed. That is what we can see when we look at you. or when we do a blood test and find out what kind of hemoglobin you have, or something like that. Your genotype is the combination of alleles, that is capital G or lowercase w or whatever, that you got from mom and dad. Your genotype will always be 2 letters because you got one from mom and one from dad. When writing genotypes, the dominant allele always gets expressed big and strong, so it gets a capital letter. The recessive allele only gets expressed when there's no dominant around, so it gets a lowercase letter. Your phenotype can be predicted from your genotype really easily. If you have at least one dominant allele, you show the dominant phenotype. So big G, little g is green. Big G is also green. If not, if you only have the recessive allele, sorry, big G, little w, you're white. If you don't have the dominant allele around, you show the recessive phenotype. It doesn't work so well going from phenotype to genotype, because you see we can get green 2 different ways. So if we know dad has brown eyes and mom has blue eyes, mom's easy. She has to be 2 little b's because that's the only way to get blue eyes. But dad could be 2 big B's or 1 of each. We call that homozygous or heterozygous. Okay, so last question. How do we predict the chance that the first baby will have blue eyes? Well, mom, we just decided, has to be little b. Dad could be homozygous or heterozygous. So let's pick the homozygous case first. So mom is always going to make the 2 possible kinds of oocytes she's going to make are both blue eyes. Dad, if we take the homozygous case, is always going to make sperm that code for brown eyes. When we cross 2 of these together, we're always going to get 1 of each. And the phenotype for that is what? Brown. If you have at least 1 capital B, you're brown. So all of their kids are going to have brown eyes. Nice and boring. But what about if dad is heterozygous? Then dad's going to make half of his sperm with brown genes and half with blue. So we'll get big B's in that row, little b's in this row. What % of the kids is going to have blue eyes? About half. Okay? Or a better way to put it is, anytime you have a kid, there's a 50% chance that they'll have brown eyes and a 50% chance that they'll have blue eyes. You actually could have 3 brown-eyed kids in a row from these parents if the random mix matching of genes just happens to work out that way, but it's unlikely. Last situation, what if mom and dad both have brown eyes? Okay, is there still a chance of having a blue-eyed kid? Or does a blue-eyed kid mean mom has been messing around with the mailman? Okay. When we cross these, 1/4 of the kids are solidly brown eyes. 1/4 of the kids, no, 2/4 of the kids, half of the kids, are heterozygous. But 1/4 of the kids have blue eyes. So that's how genes supposedly skip a generation, if you've heard that phrase. Oh, that trait's in my family, but it always skips a generation. They're talking about a recessive trait that can do this. And in fact, it usually doesn't work out this neatly, but I have 4 boys, and my wife and I both have brown eyes, and we have 1 blue-eyed kid and 3 brown-eyed kids. So it
Did you get a DNA test?
worked out.
Just kidding.
Worked out exactly right. As it happens, our mailman was a male woman, so no question there. So simple genetic disorders, real easy. If dad is a carrier for sickle cell anemia, that means he's got 1 normal trait and 1 sickle cell trait. Mom has 1 normal and 1 sickle cell. We cross it, we get 1 completely healthy kid, 2 carriers, and 1 completely sick kid. Prior to modern medicine, this kid dies fairly early in childhood. So why then is this disease at all prevalent if 1/4 of the kids of those parents are going to die? You'd think those traits would die out of the population, but it turns out that it's very prevalent in people from sub-Saharan African ancestry. They did some research into this, and they found that malaria, that people who have 1 copy of the sickle cell gene are immune to malaria. So if there's a big malaria outbreak in the area, this kid dies too. But these 2, the 2 carriers, survive. So every time there was a malaria outbreak in sub-Saharan Africa, you had an enrichment of the population for people who carried sickle cell disease. And so that is still prevalent today in people from that genetic origin. They think this is probably true of a lot of genetic disorders, cystic fibrosis, Tay-Sachs disease. Some of these things probably had a protective effect against something way back when, and if we did enough research, we'd probably find out what that was. But today they don't help us much. They just hurt. Blood types. Blood types are interesting because there are 2 dominant genes instead of just 1. A is dominant, B is dominant. You have both of them, they become co-dominant, and you get type AB. So if we had a dad who was AB, let me do red because it's blood. If we had a dad who was type AB and a mom who was type O, which is recessive, then we'd get either A kids or B kids, but never both. No kids who were both A and B. This is sometimes used as a paternity testing thing, or it used to be before reliable genetic tests. And it turns out that it's really lousy at that, because if you have a mom who's type A and a dad who's type B, and he's trying to say, well, the kid's type A, so he's obviously not my kid. From this pairing, you can get every single blood type. So there's no way to tell whose kid that is. The last one I want to talk about real quickly is a sex-linked disorder. Okay. Let's say dad is colorblind, meaning he's got the lowercase x or x colorblind, and he's got a Y. Mom had a colorblind dad, so she's got X and little x, X colorblind. Match those up. Big X, little x, little x, little x, YX, YX. From this pairing, you have equal shot of a boy being colorblind or a girl being colorblind. That's not the usual situation. Usually what happens is you have a dad who is not colorblind and a mom who is not colorblind, but you crisscross them and all the girls are fine. But half of the boys are colorblind because Y acts as a recessive gene in this case, so it has no protective effect against color blindness. So color blindness is associated with being male. It's much more likely to have colorblind boys than colorblind girls. Okay, so that concludes the genetic basics.